This is a review of Oxytocin Modulates Social Value Representations in the Amygdala by Yunze Liu et al., presented by me, Alan Blaha. Humans live in a very social environment. Decisions that we make need to take into consideration the cost and benefit of both ourselves and others. In order to understand social reciprocity and social decisions, it is crucial to understand the neural codes. Previous studies show that most people are never 100% selfish or altruistic. There are two main groups of people: individualists and prosocials. Those who prefer allocations considering the interests of both self and other, who often seek to minimize the self-other difference, are prosocials, while those who have more selfish preferences tend to maximize resources for themselves and prefer self-centered allocations. This is a clear personality difference in people, but the difference is not as clear in neural connections and pathways. This experiment looks at neural computations and representations that are related to social decision-making. The amygdala, which has oxytocin and dopamine receptors, has strong implications in social cognition and decision-making, is a prominent candidate to encode deviations from social reference point. The amygdala is known to play a role to encode error signals, which represent differences between expectation and outcome. Prosocials and individualists who have different social reference points would be expected to also have varying neural substrates for social value representations. For prosocials, deviation from the normal social reference point could show deviation from their normal social principles, such as inequity aversion. Inequity aversion is related to the amygdala. On the other hand, individualists who deviate from their normal self-interest, which guides their decision-making, could represent deviation by conflict in self-interest. The lateral prefrontal regions that are associated with the inhibition of self-interest and self-other allocations could show change. They will be looking at lateral orbitofrontal cortex and dorsolateral prefrontal cortex. Social behavior has also been shown to be regulated by the neuromodulatory regulation of neural circuits. Oxytocin is a good candidate for this because it plays an important role in social interaction and social decision-making. It plays a role in social motivation, increasing trust and cooperation among groups, and reduces social distance. The role of how and why oxytocin modulates social preference is unexplored. They predicted the differential impacts of oxytocin on regulating prosocial behavior between both individualists and prosocials by increasing prosociality in individualists by an amplification of the amygdala's social value representation. In the experiment, they tested whether intranasal administration of oxytocin differentially modulates the neural representation of social values. Using an fMRI imaging, they set up a double-blind, placebo-controlled between subjects design. First, they had to determine if the test subjects were prosocial or individualists. To do this, they had to show their social reference model to determine who was which. Oxytocin increased prosociality in individualists and not prosocials. These findings are replicated in two additional behavioral experiments that also used fMRI analysis. We'll get to more on that later. Oxytocin amplified neural representations of social values in the amygdala in individualists. These findings suggest that there's a link between oxytocin and prosociality by seeing the modulation of social value representations in the amygdala through fMRI. They invited 282 individuals to a behavioral session to identify the individuals as prosocial or individualists. After they found that 127 people would be considered one or the other. The individuals who were tested were administered with oxytocin or a placebo, and they performed a monetary outcomes for both themselves and other individuals being tested. The participants were presented with monetary outcome pairs to show specific amounts of money received by themselves and another player. The plus sign indicated a gain, and the minus sign indicated a loss. They had 3 seconds to rate their preferences from 1 to 4 for each monetary outcome pair. This was followed by 1 to 5 seconds of jittered intertrial interval. Their preference defined an angle theta, which samples a space -90 degrees to 180 degrees. The scores were plotted against monetary allocations for the self and other. Their results show that the representation of social value is a relational map which encodes the distance between potential values for oneself and others on the same coordinate system. This representation can show how humans interact with one another and how we react to perceived unfairness. Through their experiment, they proved that the social value representations are constructed in relation with the individual's specific social preferences. In prosocials, the social value distance is encoded in the amygdala. The higher their prosocial behavior, the greater the amygdala response. The results show how social value contributes to prosocial decision-making. Their study suggests that the amygdala encodes the difference between potential and preferred allocations, similar to dopamine firing representing reward prediction. They found that the trial-by-trial amygdala activity has a negative correlation with the desirability of potential self-other allocations. There is evidence that social value distance is also encoded in the ventral striatum in prosocials. The social value distance in individualists is represented in the lateral orbitofrontal cortex, which may reflect coding scheme for social values. This is consistent in other studies that looked at LOFC function. Unlike LOFC, the dorsolateral prefrontal cortex did not have any significant social value distance representation in either individualists or prosocials. The data suggests that individuals perceive deviations from their preferred allocation mainly as conflicts with self-interest. Oxytocin seems to have a role in facilitating social approach and to increase the salience of social cues and promoting adaptive social behavior. Individualists tend to focus more on self-interest, but with oxytocin, they are more likely to increase prosociality by shifting reference points to a more prosocial allocations. This may occur by the amplification of amygdala representation of social value distance. This is supported by other studies which show the amygdala plays a critical role in allocating attention to other people and integrating social information. Although oxytocin was shown to have an impact on individualists, it failed to have a significant impact on already pro-socialists. They found that even though oxytocin increases individualists' prosociality, prosocials still show higher amounts of prosocial behavior than individualists who are on oxytocin. The overall experimental results reveal a neural mechanism that underlies social value representations. This shows new insights into the process of human social decision-making. Oxytocin adaptively modulates social value representations in the amygdala and show there is a fundamental role in oxytocin in social decision-making. These insights and identification of selective effects of oxytocin on prosociality in individualists may be able to help regulate and treat a series of neuropsychiatric disorder with a social deficits, such as autism and sociopathy. Being able to help regulate neuropsychiatric disorders would be extremely beneficial. Being able to isolate the use of oxytocin to help this is still far from being perfected, but does not seem too far from being accomplished. More experimentation needs to be done to try to isolate the specific impacts of oxytocin, although this experiment was a good starting point. If I were to be the one performing this experiment, I would have tested a wider range of brain areas in prosocials. After finding that the behavior of prosocials was not impacted by the oxytocin, I would have gone into more detail on why that is and what causes the prosocial behavior in the first place. The idea that there are two groups of people, individualists and prosocials, seemed obvious to me, but the fact that their neural activity in social environments is different is fascinating. Their neural activity is directly correlated with their personality, or maybe their personality is because of their neural activity. This has many other implications. Any childhood behavior disorder, whether it be attention deficit, obsessive compulsive, oppositional defiant, etc., can be better regulated with specific isolations of oxytocin. In order for this to work, neuroscientists need to be able to isolate the specific brain regions that would need the extra boost in oxytocin.